Good evening, everyone. So, we're gonna start in 5 minutes as usual so that more people can join in. Okay? इंसान की तरीके से नहीं होती फिर इंसान को ज़्यादा पावर लग के मिलेगी So I guess we can start and people can join in between. So yeah, so we were in the last class, what we have done as usual. So we revised most of the things which we covered, and then we were supposed to revise from here. So before we start our revision and then we add new topics as well, this is our last second class. So most of the immunology and most of the topics, most of the say building blocks of immunology have already been introduced to you guys. Okay, so it's a huge subject, it's a vast subject. It has it has ranges from genetic engineering to cell biology to molecular biology. Every single aspect is there in immunology. Okay, so chances are that students do get overwhelmed with this subject. So the approach which worked for me, and I hope that it works for you as well, is follow the sequence through which the lectures were introduced to you. So firstly it was just the summary, overall summary. Then we went into types of immunities, then we went into components of immunity, then we went into effector molecules of those components, then we digged further into how these components are cross talking with each other, and then how exactly these components are being made, right? So if you take this approach, chances are the subject will be little less overwhelming, right? So in the first go, for sure it will be overwhelming, but gradually if you keep repeating and learning and relearning the similar concepts, it becomes like a story. Okay, so if you want to revise it on your own, so how you can do is, you can basically, so you remember we introduced the inflammation, and how we learned about the inflammation is, oh, that there is a infiltration, or there is a breakage of the primary barrier, then the microbe has entered, and then how each and every cell component is playing role in making sure that the inflammation takes place, and the adaptive immunity knows while the inflammation is taking place that there is a microbe, there is a pathogen who has entered our system, and we need to take appropriate steps. Okay, so if you try to make it as a story since the first incident of it, then it will be easier for you to catch up with the subject, first thing. Second, and stay connected with the subject. Okay, so this is just a basic suggestion of how you can make this subject more entertaining, more enjoying, and at the same time that you are learning. Okay, so yeah, so you know about the complement system, right? So what exactly is the complement system? The complement system is a network of circulating and membrane bound proteins, crucial for host defense against microbes and for antibody mediated tissue injury. Its name comes from the fact that it complements the action of antibodies in destroying the cells or taking care of the infection. It can be activated in majorly two ways: innate immunity and adaptive immunity. When we talk about innate immunity, so when the complement system is getting activated, and if it is innate immunity, then what we need for the activation of this complement system, we only need the microbe which is not bound with the antibodies. Okay, when we talk about adaptive immunity and how adaptive immunity is activating the complement system, so the basic requirement for this to activate the complement system is the antibodies which are bound to the microbial surface. Activation proceeds through a cascade of proteolytic cleavages, producing effector molecules that eliminate microbes by mechanisms such as cell lysis and opsonization. Like other enzymatic cascades, this process always allows amplification, where small initial signal generates a large downstream responses. So this similar behavior we have seen in signaling as well, right? So few ligands will come and bound to the surface and surface receptor, and then a lot of downstream signalings, downstream molecules, secondary messengers will be there, which are amplifying the ligand which just came and bound to the receptor. Okay. Importantly, activated complement proteins bind covalently to the microbial or target cell surface, localizing the attack to the right place. To prevent harm to self, host cells have regulatory proteins that inhibit activation and deposition of complement components, thereby avoiding damage to healthy tissue. Okay, we have already discussed this, but as it has been 2 to 3 weeks since we have actually understood about the complement system, I'm not gonna cut short the complement system part. Okay.
Immune response and plays a major role in the killing and clearance of invading pathogens, including bacteria, viruses, fungi, and parasites. The system is composed of over 30 proteins, most of which are found in serum, while the remaining proteins are membrane-bound proteins and receptors. On activation, complement proteins that participate in the activation of either the classical or alternative pathways of complement interact in a highly specific enzymatic cascade and generate proteolytic fragments that mediate the numerous biological functions of the complement system. In this video, we will focus on the activation and biological functions of complement. Activation of the classical pathway. The classical pathway is typically activated by antigen-antibody complexes. Although there are many other activators of this pathway, only two isotypes of antibody, IgM and, as shown here, IgG, will activate the classical pathway. While one pentameric IgM antibody will activate the classical pathway, a minimum of two IgG antibodies in close proximity are required. In either case, antibody must be bound to antigen in order to activate the classical pathway. The classical pathway is activated when C1, the first protein in the pathway, binds to the Fc portion of the antibody. C1 is a large macromolecule composed of a C1q molecule and two molecules each of C1r and C1s. It's the globular head portion of the C1q that binds to the Fc binding site on the antibody. This binding causes a conformational change in C1q and subsequent autocatalytic conversion of C1r to an active serine protease. Activated C1r cleaves C1s to an active serine protease. Activation of C1 by binding antibody is also termed complement fixation. The next step in the activation of the classical pathway is the cleavage of C4 by the activated C1 molecule. C4 is cleaved by the activated C1s molecule in C1 into two proteolytic fragments, a small peptide called C4a and a larger fragment, C4b. C4b attaches covalently to the antigen-antibody complex and has a binding site for C2, the next protein cleaved in the classical pathway. C2 is also cleaved into two fragments, and the resulting C2a fragment binds to C4b. This bimolecular complex of C4b2a is a C3 convertase of the classical pathway and serves to cleave C3 through the enzymatic activity in the C2a portion of the complex. Classical pathway C3 convertase. The C3 molecule is cleaved by the C3 convertase into C3a and C3b. The C3a fragment has potent biological activities, which will be discussed shortly. The C3b fragment can covalently attach to the C3 convertase, resulting in a trimolecular complex consisting of C4b, 2a, 3b. This multimolecular complex is called the C5 convertase of the classical pathway and is specific for cleaving C5. classical pathway C5 convertase. The C5 molecule is cleaved by the C5 convertase into C5a and C5b, both of which mediate important host defense functions of the complement system. Both C3a and C5a are potent chemoattractants. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. The terminal complement pathway, or formation of the membrane attack complex. One of the best-known host defense functions of the complement system is its ability to lyse many bacteria, envelope viruses, and nucleated cells. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis. Activation of the alternative pathway. The alternative pathway of complement is activated by LPS and other lipopolysaccharides found on the surface of invading pathogens. The alternative pathway is always active at a very low level. This activation is due to the spontaneous hydrolysis of an internal thiol ester bond in C3. Hydrolyzed C3, called C3 water, has a binding site for factor B, another protein in the alternative pathway. Factor B binds to C3 water and becomes a substrate for cleavage by factor D, denoted here as FD. Factor D cleaves factor B into two fragments, BA and BB. The enzymatically active BB fragment remains associated with C3 water, and this bimolecular complex is the alternative pathway C3 initiation convertase. Alternative pathway C3 initiation convertase. This initiation convertase cleaves C3 into two fragments, C3a and C3b. The newly generated C3b can covalently attach to nearby surfaces, just as C4b does in the classical pathway. In the absence of an appropriate activator for the alternative pathway, complement regulatory proteins block further activation of the pathway. However, C3b, on interacting with an activating surface, such as a bacterial cell, will bind factor B, which will be cleaved by factor D, resulting in the formation of the alternative pathway C3 convertase of C3b-BB. Alternative pathway C3 convertase. The C3 convertase of the alternative pathway cleaves C3 to C3a and C3b. Some of the C3b generated by this cleavage does not interact with the activating surface and remains soluble. Some of the C3b generated will covalently interact with the activating surface and start the formation of additional alternative pathway convertases, thus rapidly amplifying activation of complement through this pathway. The surface of an activator, such as a bacterial cell, can rapidly be covered with many thousands of C3b molecules in a few minutes as a result of this amplifying effect. And, as with the classical pathway, some of the C3b generated will interact with the C3 convertase itself, resulting in the formation of a trimolecular complex of two C3b molecules and BB. This multimolecular complex is called the C5 convertase of the alternative pathway and is specific for cleaving C5. The C3a and C5a fragments generated by activation of the alternative pathway mediate the same functions of chemoattracting and activating granulocytes and phagocytic cells. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. Activation of the alternative pathway results in the formation of the membrane attack complex, leading to the lysis of the invading pathogen. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis.
Right? Correct. So why I'm focusing a little bit on visual aids for complement system is that if you sit and you just try to mug up each and every single component of complement system, chances are that you might not say understand or won't be able to remember these things for long term. So if you see them as a story, that okay, we have a microbe who is coated with antibody, then which pathway is being activated, then you know the whole cascade, because in like every single complement system, it will ultimately merge to C5 convertase, right? It's just their initiation is different. So when you look at those initiation, then learn it as a story, that if antibody is present and it is attached to the microbe, which pathway is being activated. If the antibody is not so antigen bound antibody is not present, only antigen is present, then how the system, the initiation of that system is taking place, because at the end of the day the end product is same. And what is the end product? The end product is making a pore in the membrane of the cell so that it can be cleared out from the system. Okay, so let's
First
see
line of
another
defense.
summarization.
Now it's
of
time to talk
complement
about
system.
another component of innate immunity called the complement system. Late 17th century, it was well understood that blood from various laboratory animals had bacterial killing properties in vitro. However, if the blood was heated sufficiently before the experiment, it could no longer kill bacteria. This phenomenon became even more puzzling when scientists transferred heat-treated blood from guinea pigs who had recovered from cholera infection into new animals. The new guinea pigs were still protected from cholera infection even though the heat-treated blood couldn't kill bacteria in vitro. In the 1890s, the Belgian scientist Jules Bordet suggested that there were two distinct protective components in the blood: the heat-stable component, which protected the guinea pigs in vivo, and the heat-labile component, which lost in vitro killing capacity when heated. In 1899, the immunologist Paul Ehrlich named the heat-labile component complement because it complemented the other heat-stable aspect of the immune system. The heat-stable component was later discovered to be antibodies, which we will discuss at great length later in the series. Complement is an evolutionarily ancient aspect of the innate immune system, and homologs of mammalian complement proteins have even been found in invertebrates like starfish, sea urchins, mosquitoes, and sea squirts, which illustrates that it's been around for a long time. So, what exactly is complement? The mammalian complement system is made up of several proteins that circulate in the blood. Many of these proteins are enzymes called zymogens, which means that they normally exist in an inactive state and must undergo a biochemical change before they can be enzymatically active. When one protein gets activated, it cleaves and activates the next protein in the sequence, which in turn cleaves the next. The result is a molecular domino effect that leads to immune system engagement and pathogen killing. The nomenclature for the complement system can be a bit complicated, even for immunologists, so let's break it down a bit. There are 9 main complement proteins, which are named with the capital letter C and then a number. The tricky part is that the number comes from the order in which the proteins were discovered, not the order they appear in the complement cascade, so keep this in mind moving forward. When a complement protein is cleaved, the cleavage products are given a lowercase a or b after the number. The A fragment is always the smaller fragment, an anaphylatoxin, while the B fragment is the larger binding portion, except in the case of C2. For example, when C3 is cleaved, it is cleaved into C3A and C3B, with C3A being the smaller product.
This is how the brain of a procrastinator.
You will soon see many of these cleavage fragments complex together to form enzymes or other functional proteins. To designate which complement proteins make up a complex, the names of the proteins are strung along in the order in which they bind. For example, the classical C3 convertase is called C4b2A because it is made up of C4b and C2A. When C3b binds to this complex to create a C5 convertase, the complex becomes C4b2A3B. As a quick note, some sources have recently begun swapping the designation for C2 so that the C2A and C2B fragments match the rest of the system, which would make the C3 convertase C4b2B instead. But this convention, as logical as it may be, does not seem to have been officially adopted, so it may be important to be aware of this inconsistency within the field, depending on your specific purposes in learning immunology. Now that we understand the system of nomenclature, let's learn about how the complement system actually works. Complement can be activated through one of 3 distinct pathways. Each of these pathways converges at the step of C3 cleavage, which kicks off the most important effector functions of the complement system. We'll discuss the activation of all 3 pathways first, and then circle back to what happens after C3 is cleaved. The classical pathway was the first complement pathway to be discovered. The classical pathway begins with a protein complex called C1. C1 is made up of 3 distinct subunits: C1q, C1r, and C1s. C1r and C1s are proteases, which are enzymes that cleave proteins, but they are inactive during normal conditions. During an infection, circulating C1q can bind directly to some bacterial surfaces, to antibodies bound to a pathogen, or to an acute phase protein called C-reactive protein that also binds to bacterial surfaces. C1q binding causes a conformational change, resulting in autocatalytic activation of C1r. Activated C1r then cleaves.
Okay, so what do we mean by autocatalytic activation? So if you guys have little bit of idea regarding the protein structures, so imagine this protein didn't have any catalytic function to begin with, right? Once it get attached to the required protein or ligand, or say any small molecule, then it changes its form from being non-activated to activated. So what exactly that means? It means that there is a structural change in the protein which leads to the access of that opening or that hidden structure when it is in the native form, and when the protein gets attached to its complement commodity or small molecule. Due to this structural change, now the structure which was hidden, it is exposed to the solvent. Once it is exposed to the solvent, then it can do its function. So in this case, it will be the catalytic function. Catalytic basically means breaking of things, right? So if and it depends upon which type of cleavage it is. So there are serine cleavages, there are enzymes which cleave which cleave protein at wherever there is tyrosine residue is present. Okay, so all these specificities comes only when the structure is accessible to other proteins or small molecules, and this is a general principle for any protein. So when we say that how come the snake venom, which is like deadly to us, is not deadly to the snake itself, or say the bee which stung us is not suffering from that pain and all when it is inside the bee itself. Okay, so all these things revolves around pro activated state, which means when it is not activated, and then activated state. And the major thing which occurs from pro form, converting pro form to active form, 80 to 90% of the times it is structural change only. Okay, so you have seen a small example of structural importance in the immunology. If you remember IgM antibody, so IgM antibody is both membrane bound as well as soluble, right? So when the transmembrane domain is present and it is present in the on the cell surface, it is membrane bound. When that transmembrane domain or the structure which is hinging the antibody to the cell surface, it's not there, then it is in the soluble form. So the occurrence of transmembrane domain, if it is present, then it is showing one type of function. If it is not present, that is structural change, then it is showing another function. Okay, so this was just a basic information.
And activates C1s. Once C1s is activated, it cleaves circulating complement proteins C4 and C2. C4b covalently binds to the pathogen's surface or to pathogen-bound antibodies, and C2a binds to C4b, forming the C4b2a complex. This complex is known as the C3 convertase, and it cleaves circulating C3 into C3a and C3b. Although the classical pathway of complement activation was the first to be discovered, it is likely the youngest evolutionarily. After all, it often relies on antibody recognition of pathogens, and antibodies are only found in jawed vertebrates. Since complement can be found in lower order organisms, there must be other ways for complement to be activated. One of these pathways is called the lectin pathway. Lectins are proteins that can recognize and bind to very specific carbohydrate groups. While some lectins play homeostatic roles within the host, others act as pattern recognition receptors, detecting arrangements of sugar molecules that are only found on pathogens, such as those found on bacterial cell walls or adorning yeast cell surface proteins. So far, we know of 4 lectins that activate complement after binding to microbial sugars. These include mannose-binding lectin, or MBL, and phycolins 1, 2, and 3. Mannose-binding lectin is made in the liver and circulates in the bloodstream. It is a multimeric protein with multiple binding sites that recognize the sugar groups fucose, mannose, and N-acetylglucosamine. These sugar groups are commonly exposed on microbial polysaccharides, but not on host cells. The phycolins are structurally similar to mannose-binding lectin, except that they bind acetylated sugars instead of mannose groups. Now, here's how lectin binding initiates complement. In the circulation, mannose-binding lectin, or one of the phycolins, complexes with the proteins MASP1 and MASP2, which are MBL-associated serine proteases.
आपका सहयोग, आपका दान, इनके सब
Proteases are inactive until the lectin binds to the sugars on a pathogen's surface. At this point, a conformational change activates MASP2, allowing it to cleave and activate another molecule of MASP2, which can then cleave the complement proteins C4 and C2. C4b complexes with C2a to form the C3 convertase, and here the pathway converges with the classical pathway. The final pathway of complement activation is known as the alternative pathway because it was discovered after the classical pathway. Although it is called alternative, it is probably much older evolutionarily than the classical pathway. The biggest difference between the alternative pathway and the other two is that it utilizes a distinct C3 convertase. This convertase consists of C3b bound to a cleaved fragment of a plasma protein called factor B. When C3b is bound to the surface of a pathogen, circulating factor B can bind. This allows another plasma protein called factor D to cleave factor B, leaving behind the rather confusingly named C3bBB. This is the alternative pathway C3 convertase. You might be wondering how one of the subunits of this C3 convertase is C3b, since C3b can only be generated by a C3 convertase. Remember that C3b can be generated by the lectin or classical pathways. Additionally, spontaneous hydrolysis in C3 can lead to a short-lived intermediate convertase that can also generate C3b. So what happens after the generation of the C3 convertase? Remember, for the classical and lectin pathways, the C3 convertase consists of C4b2a, and for the alternative pathway, it is C3bBB. C3 is the most abundant complement protein in the serum, even with a meager concentration of 1.2 milligrams per milliliter, and because of this, C3 has incredible capacity to amplify early complement events. Additionally, C3b can create more of itself by utilizing the alternative pathway. Remember that both kinds of C3 convertases are bound to the pathogen surface. When it is exposed to circulating C3, the convertase cleaves the C3 protein into two parts, C3a, the smaller part, and C3b, the larger. C3a is released into the circulation, where it recruits neutrophils and other phagocytes to the site of infection. C3b, on the other hand, can do one of two things. The first thing is that many molecules of C3b can bind to the surface of the pathogen, coating it in a process called opsonization. While opsonization doesn't directly destroy the pathogen, it marks it for destruction by phagocytes like macrophages. Phagocytes have receptors that recognize C3b coating the surface of a pathogen and will phagocytose objects or organisms coated in C3b. Opsonization is especially effective when the pathogen is also coated in antigen-specific antibody, as phagocytes also have receptors that recognize bound antibody. The second thing C3b can do is bind to the existing C3 convertase, creating a C5 convertase. As you might imagine, once the C5 convertase is formed, it can cleave C5. Like C3a, C5a is released into the circulation to recruit phagocytes. Meanwhile, C5b nucleates the membrane attack complex, a complex of complement proteins that form pores in pathogen cell membranes, causing the pathogen to lyse. Here's how this works. C5b binds to circulating C6 to form C5b6. C5b6 then binds to C7. Binding causes a conformational change in C7, which exposes hydrophobic residues, allowing C5b67 to insert into the pathogen's cell membrane. C8 then binds the complex, which also causes a conformational change, allowing C8 to insert into the pathogen's plasma membrane. Finally, 10 to 16 molecules of the terminal complement protein C9 polymerize within the cell membrane to form a pore that allows water and solutes to flow out of the pathogen, leading to death. This is possible because while the outer surface of the pore is hydrophobic, allowing it to insert into the plasma membrane, the inside surface is hydrophilic, allowing polar molecules like water and ions to travel freely through it. Although the membrane attack complex is able to kill many kinds of bacteria and parasites, it is especially important for controlling bacterial infection with Neisseria species and may actually be dispensable for controlling other infections. The evidence for this is that genetic defects in the terminal complement proteins C5 to C9 do not result in increased susceptibility to any known pathogens except Neisseria.
If you're an executive who wants to communicate your ideas as powerful lead, you must join our community.
Although C3A and C5A are sometimes forgotten because they don't contribute directly to pathogen clearance, they still have potent biological effects. They can induce contraction of the smooth muscle cells lining blood vessels, increase vascular permeability, and signal endothelial cells to increase expression of adhesion molecules necessary for immune cells to leave the bloodstream and enter an infected tissue. While these effects are essential for recruiting phagocytes to the site of infection, when levels are too high, C3A and C5A can lead to anaphylactic shock. This is why they are often referred to as anaphylatoxins. Because of how quickly the complement cascade can proceed and be amplified, and because of how potent the effects are, it is extremely important to be able to regulate this response to keep it from getting out of control or from attacking host cells. There are several ways the complement system can be regulated. One major way is that all the enzymes involved in the complement system, except factor D, are zymogens, meaning they must be activated before they can perform their enzymatic functions. Another way to maintain control of the complement system is that all of the enzymatic activity occurs at the pathogen's surface, which prevents active enzymes from circulating around in the bloodstream. After cleavage, both C4b and C3b have a highly reactive exposed thioester that allows them to covalently attach to any adjacent carbohydrate or protein, which is usually the pathogen's surface or an antibody that has already attached to the pathogen's surface. If they don't bond quickly, this reactive group is inactivated by hydrolysis, ensuring that host proteins aren't indiscriminately tagged by C3b. There are also specific proteins that regulate complement activation. The C1 inhibitor is a serine protease inhibitor that binds active C1R/C1s, dissociates it from CQ, and prevents it from cleaving C2 and C4. Decay accelerating factor can compete with factor B for C3b binding, preventing alternative complement activation. It can even displace bound BB, inactivating the alternative C3 convertase. Decay accelerating factor, as well as C4 binding protein and complement receptor 1, can also bind to C4b in the classical C3 convertase and displace C2a. Factor H is another plasma protein that can outcompete factor B. It preferentially binds to C3b on host cells, preventing complement activation on host cells while not interfering with binding to pathogens. It can also displace C3b from the C5 convertase. Plasma proteins like vitronectin can bind to any C5b67 complexes in the circulation, preventing them from inserting into host cell membranes and forming the membrane attack complex. Finally, protectin is a membrane-bound protein that prevents C9 from binding to the C5b678 complex, acting as yet another way to prevent complement activation from harming host cells. So that covers the basics regarding the complement system, which is just one of the many complex aspects of the immune system. Let's move forward and learn about the
I'm so sorry for the information overload, but if you guys were focused enough throughout the video aid, the chances are that whatever I'm going to discuss here will feel like just a revision, and at the end of the day we'll see this culmination and like see them together side by side, so that we can draw parallels as well as we can draw things which are different among these pathways. As I've mentioned again and again, the components are same. How they are assembling, that is the only difference. So without worrying too much about, oh, a lot of things have to be remembered, it's true, but if you remember the initiation part of each pathway, it will be very easy, it will be very flowy, because then the components are same. Okay, and in these type of things visual aid does help a lot, because now you can put images, you can put like reference structure to each component, which will help you in understanding and remembering the pathway as well. Okay, so by now you know there are majorly three pathways of complement activation. The alternative and the lectin pathways are initiated by microbes in the absence of antibody, and this is one of the reason we say that classical pathway, like the term classical pathway, is associated, because it is only initiated when antibody is there, and antibodies are not present throughout the throughout the domains of life, all the domains of life. Okay, so still these pathways exist and they recognize the microbe itself, which means the immunity, the complement system is older than the adaptive immunity. Okay, and the classical pathway is initiated by certain isotypes of antibodies attached to antigens. Several proteins in each pathway interact in a precise sequence. The most abundant complement protein in the plasma, C3, plays a central role in all three pathways. The early steps of all three pathways function to generate a large number of functionally active fragments of C3 bound to the microbe or cell where the complement pathway was initiated. By convention, the smaller prototype will be called A and the larger prototype will be called B, except for the C2. Okay, so first we'll see the little bit information about the alternative pathway. The alternative pathway of complement activation is triggered by spontaneous hydrolysis of C3 in plasma at a low level. The breakdown product of C3 are unstable and, in the absence of infection, are rapidly degraded and lost. However, when a breakdown product of C3 hydrolysis, called C3b, is deposited on the surface of a microbe, it forms stable covalent bonds with the microbial protein or polysaccharides. The microbe-bound C3b binds another protein called factor B, which is then cleaved by a plasma protease called factor D to generate the BB fragment. Now, I really hope that when I am going through the description of pathways, you can actually imagine these components breaking and coming together, right? This fragment remains attached to C3b and the C3b-BB complex functions as a proteolytic enzyme called the alternative pathway C3 convertase that breaks down more C3s. The C3 convertase is stabilized by properdin, a positive regulator of the complement system. As a result of the enzymatic activity of the C3 convertase, many more C3b and C3b-BB molecules are produced and become attached to the microbe. Some of the C3b-BB molecules bind an additional C3b molecule, and the resulting C3b-BB3b complexes function as a C5 convertase now, okay, to cleave the complement protein C5 and initiate the late steps of the complement activation, that is formation of the membrane attack complex. While the classical pathway of complement activation is triggered when IgM or certain subclass of IgG binds to antigen. As a result of this binding, adjacent Fc region of the antibodies become accessible to and bind the C1 complement protein, which is made up of a binding component called C1q and two proteases called C1r and C1s. The attached C1 becomes enzymatically active. See, earlier it was not, but when certain components come and bind to the protein which already exists, it will make the enzymatic site available to the surface, and then it becomes active. Again, a structural change, okay, resulting in the sequential cleavage of two proteins, C4 and C2. One of the C4 fragments that is generated, C4b, becomes covalently attached to the antibody or to the microbial surface where the antibody is bound, and then binds C2, which is cleaved by active C1 to yield the C4b2a complex. This complex is the classical pathways C3 convertases, which functions to break down C3, and the C3b that is generated again becomes attached to the microbe. Some of the C3b binds to the C4b2a complex, and the resultant C4b2a3b complex functions as a C5 convertase, which cleaves the C5 complement protein. So in alternative pathway, C5 convertases is called C3b-BB3b, while in classical pathway, C5 convertases is called C4b2a3b. Okay. And last but not the least, the lectin pathway of complement activation is initiated not by antibodies but by the binding of circulating lectins such as plasma mannose binding lectins or ficolins to microbial polysaccharides. Serine proteases structurally relate to C1s of the classical pathway, are associated with these lectins and serve to activate C4. The subsequent steps are the same as in the classical pathway. Now we'll see the same things which we saw in the visual aid, which we read through in a subjective way. Now we'll see in the parallel way. Okay. So this is the alternative pathway, this is the classical pathway, this is the lectin pathway. So major events which takes place in the complement system activation are binding of complement proteins to microbial cell surface or antibody, formation of C3 convertase, cleavage of C3, formation of C5 convertases, and finally cleavage of C5. So how these steps are taking place in different pathways, we'll see this. So in the alternative pathway, the microbe will have bound C3b molecule entity covalently, which will be further bound with BB component. This C3b-BB will further bind to C3b, which will be called as C3 convertases. Now this along with another C3 molecule, that is C3b-BB C3b molecule, is called C5 convertases. And when it is called C5 convertases, which means it will basically break the C5 complement protein, and the C5a and C5b have different functions. Okay, so here the initiation is made by covalently linked C3b component to the microbial surface in alternative pathway. In classical pathway, what will happen? A microbe along with antibody is needed. So a microbe is there, IgM antibody is there, IgM antibody's Fc part will be recognized by C1 protein, which is further bound with C4 and C2. This component has the ability to cleave C2. Okay, then it is when they both are joined, like C4b and 2a, they are called C3 convertases. When a molecule of C3b binds to this component of C4b2a3b, this is called C5 convertases, and its function will be same, that it will break down C5 complement protein. When we look at lectin pathway, it will recognize the lectins on the surface of the microbe by C4 and C2. So here the C3 convertases of classical pathway and lectin pathway are same. So as you can see, it's just the initiation of how the complement is being activated is different between classical pathway and lectin pathway. All other steps are same. While in alternative pathway, the initiation as well as the progression of C5 convertases is different. So remembering these both pathways in which microbe is recognized by the circulating complement proteins will follow similar path. Okay. Now, what are the major functions of these complement proteins? So the C3, if we say, so as we said that C3 is important, right? So once C3 is being broken down, then it is a major component of C3 convertases. Okay, it makes C5 convertases whenever there is a part of C3 protein in it. Okay, so the function of C3 protein is C3b binds to the surface of a microbes, where it functions as an opsonin and as a component of C3 and C5 convertases. C3a stimulates inflammation. So here, as you can see, that most of the components are B only. Okay, except complement part 2, like complement protein 2. Only the B components are making this convertases or enzymes. Then what is happening with the A components? So for example, here C5a, C2b, C3a, what are these components doing? So they also have separate function. But here we are discussing majorly about the B components, because they are the ones which are making the enzyme convertases and then finally the membrane attack complex. Okay, so out of that A components, one of the function of C3a component is to stimulate the inflammation, and you know how the inflammation takes place. Okay, factor B. So BB is a serine proteases. So what do we mean by serine proteases? As I've mentioned like while I was describing how the enzymes function. So serine proteases means that it will show its proteases activity. It's an enzyme on residue wherever there is serine or with the help of serine residue. Okay, and the active enzyme of C3 and C5 convertases. Factor D, plasma serine proteases that cleaves factor B when it is bound to C3b. Properdin stabilizes C3 convertases on microbial surface. So all these components have some role to play. When we talk about classical and lectin, so as you know, both classical and lectin are same, how they are progressing once the formation of C3 convertases take place, is just their initiation is different. Okay, so in classical and lectin pathway, C1, so which has C1QR 2S2 components associated with it, so it initiates the classical pathway. where C1q binds to Fc portion of antibody. C1r and C1s are proteases that lead to C4 and C2 activation. C4b covalently binds to surface of microbes or cells where antibody is bound and complement is activated. C4b binds to C2 for cleavage by C1s. C4a stimulates the inflammation. C2a is a serine protease functioning as an active enzyme of C3 and C5 convertases. Mannose binding lectins. So initiates the lectin pathway. MBL binds to terminal mannose residues of microbial carbohydrates. MBL associated proteases activate C4 and C2 as C1r, C1s, and they do in the classical pathway. So here we are seeing a lot of proteases name, right? So what exactly is a protease? Any one of you can share in the chat. It's a generic term, right? So you guys might have heard about it, read about it. So what exactly is a proteases? Anyone? As the name suggests, right? So it's a generic convention as well, naming convention. Yeah, that was a 1 hour cutoff. Yeah, so what do we mean by, so as I was asking, what do we mean by proteases? It's a generic term. Anyone? You can type in the chat box if you want. So proteases, so basically these are the enzymes that basically digest the protein. Proteases means enzymes that are digesting protein. Now if I am saying serine proteases, so what serine protease will does? So serine protease, they are the enzymes that will cut the peptide bond. So what are peptide bonds? Peptide bonds are the bonds which are linking amino acid residues, right, to make polypeptide or peptides. So serine protease, for example, they are the enzymes that cut peptide bonds in proteins. And why serine specifically? Because while they are cutting, from where they are cutting, that site has serine always. So it's highly conserved. That is why they are called serine proteases. It's a general side information. Then when we say the later steps of complement activation, so now we have C5 convertases. Then what is the function of C5 convertases, right? So in the end, once C5 convertases is made, it will attract other complement proteins on it, such as C6, C7, C8, and multiple units, around 9 units of C9, to finally make membrane attack complex or MAC. And in the visual aid, you have seen what this pore does. This pore basically connects the internal microbial homeostatic environment to the solvent or the aqueous surrounding, which will lead to the imbalance in their ions, imbalance in their nutrients, and finally it will lead to the death of the microbe. Okay, it is just like opening so if there's a balloon, right, balloon filled with water, imagine. Okay, now if you make a hole in it, every single, like whatever is inside the balloon, it can be water, it can be air, it will come out, okay, and then at the end the balloon will shrink. Similar thing happens with the microbial cell whenever there is MAC or membrane attack complex. So again, when we were looking at the components, the properties of proteins in the later step. So we have not seen about C5, about C6, C7, C8 and C9. So what are the basic functions of these? So for C5, the C5b, we know that it is part of C5 convertases, okay. Then so C5 initiates assembly of the membrane attack complex, while C5a stimulates inflammation. Role of C6. So the component of the MAC that binds to C5b, and it is the docking site for C7. What does C7 do? So component of the MAC which binds to C5b6 and inserts into the membrane. So C7 has the property of attaching this component to the membrane, okay. Then what is C8? So it binds to C5b, C5b6, 7 and initiates the binding, or it provides a docking site for the C9 protein molecules. C9, so it binds to C5b, 6, 7 and 8 and polymerizes to form the MAC in the end, okay. So it is all sequential. So there will be components which will form C3 convertases, which will lead to more breaking of C3, which will be called as C5 convertases. C5 convertases will then get associated with C6, 7, 8 and multiple units of 9, making the membrane attack complex, and finally the function of complement system will take place, okay. So what are the major functions of complement? So now you know one is the membrane attack complex, right? Other is stimulating inflammation. Another function is opsonization and phagocytosis as well. So as the complement name itself suggests, that it is complementing the already existing innate immunity and adaptive immunity. So one way we have seen that it will directly make the attack complex and will kill the microbe. Another, we have seen that the A components will initiate the inflammation, and we know what happens in the inflammation. Another function is the opsonization and phagocytosis, which basically makes the microbe tag, or it tags the microbe, so that our system, especially the macrophages, they find these entities very easily and then they digest it, okay. So if we look into little bit more details of opsonization, so the mechanism through which opsonization occurs is majorly via C3b component, okay. So when the complement protein C3b binds covalently to the surface of microbe, it tags them for destruction, or here like destruction is little bit dramatic, you can say it tags them for effective phagocytosis, okay. Phagocytes such as neutrophils and macrophages express complement receptor type 1, which specifically recognizes C3b. Binding of C3b coated microbes to CR1 enhances phagocytosis. C3b act as an opsonin, improving the efficiency of innate immune cells to clear the infection. Second function was inflammation. So the small peptides C3a and C5a, which are not making membrane attack complex, okay, because not any except C2a, no other A components were involved in the convertase making or enzyme making of the complement system, okay. So the small peptides C3a and C5a generated during proteolysis of complement protein C3 and C5. Functions of inflammation, you know. So 5a especially does chemotaxis, that is, it will attract and recruit neutrophils and leukocytes to the place where the infection has been found. Then the leukocytes will be activated via C3a and C5a. Vascular changes, because we need more leukocytes into the place, so there will be in changes in the permeability of the blood vessels. So this all we have studied under the umbrella term of inflammation. Okay. Then when we talk about membrane attack complex, so you know this very well, that membrane attack complex basically ruptures the whole cell. Okay, that is its function. So this is the representation of this is the basic representation of the function of pathways, how they are initiated, what are the components they are making. So I'm not going to go too deep into this, because kindly check your mics. Kindly mute yourself if you don't have any question. Okay. So what was I saying? So I'm not going to go too deep into it, because we have spent enough time in the visual aid, as well as these slides will be with you. You can go through to them deeply, or you can just skim them. But once you understand this, the whole complement pathway is if you understand it, then it is one of the easiest concept in the immunology. Okay, so again I'm repeating myself, what you have to remember in the complement, it will help you in your exams as well. How they are initiated, what are the components of C3 convertases, what are the components of C5 convertases. That's it. Okay. Then the next topic which we are going to start today is, so we'll spend maybe 10 minutes so that we can do more questions, and then we can continue as usual in our next class. Okay, so the topic which we are going to discuss is called hypersensitivity. So the concept that the immune system is required for defending the host against infections has been emphasized throughout the chapter, throughout the book, like whichever you are referring to. However, immune responses are themselves capable of causing tissue injury and diseases. Injurious or pathologic immune reactions are called hypersensitivity reactions, because they reflect excessive or aberrantly directed immune responses. Hypersensitivity reactions may occur in two situations. First, responses to foreign antigens may cause tissue injury, especially if the reactions are repetitive or poorly controlled. Second, the immune responses may be directed against self or autologous antigens as a result of the failure of the tolerance. Okay, so as the name itself is suggesting, right, hypersensitivity, which means something which is aberrant, something which is occurring more than it should. Okay, so hypersensitivity reactions are classified on the basis of the peripheral immunologic mechanisms that is responsible for the tissue injury and disease. Immediate hypersensitivity, antibody specific hypersensitivity, so especially if they are IgG, and some other type of hypersensitivities. So if we talk about immediate hypersensitivity or type 1 hypersensitivity, it is usually caused by the release of mediators from the mast cells. This reaction most often depends on the production of immunoglobulin E, antibody against environmental antigen, and the binding of IgE to mast cells in various tissues. So here you remember that when we were talking about the functions of antibody, we specifically said that during hypersensitivity, or whenever there is requirement of degranulation of histamines, IgE antibody is the one which is taking place along with the mast cells. Now this is the exact thing which we are representing under the umbrella term of hypersensitivity. So antibodies such as IgG that are directed against cells or tissue antigens can damage these cells or tissues or can repair their function. impair their function, sorry. These diseases are said to be antibody mediated or type 2 hypersensitivity. Antibodies against soluble antigens in the blood may form complexes with the antigens, and the immune complexes may deposit in the blood vessel in various tissues, causing inflammation and tissue injury. Such disorders are called immune complex disease or type 3 hypersensitivity. Some diseases result from the reaction of T lymphocytes specific for self-antigen or microbes in tissues. These are called T cell mediated diseases or type 4 hypersensitivity. So here we'll see them in little bit brief or summarized way. So basically there are 4 types of hypersensitivity: type 1, type 2, type 3 and type 4. In type 1 hypersensitivity, it is also called immediate hypersensitivity, because what happens in this, so it is mast cell derived mediators, so vasoactive amines, lipid mediators, cytokines, etc. So cytokine mediated inflammation will take place, okay, along with IgE and the mast cell. In antibody mediated type 2 hypersensitivity, IgM, IgG antibodies against cell surface or extracellular matrix antigen will be there, leading to the complement and Fc receptor mediated recruitment and activation of leukocytes. So you know now that the A component will be activated, especially C5a and C3a, which will lead to this type of hypersensitivity. Opsonization and phagocytosis of cells, abnormalities in cellular function, example hormone receptor signaling. You don't need to worry about this example. Then immune complex mediated disease in type 3 hypersensitivity. So what happens in this, basically the immune complexes of circulating antigens and IgM or IgG antibodies deposits in the vascular basement membrane, and this leads to the activation of complement system. So tell me which type of complement system will be activated here when there is antigen antibody complex is present. So you can like give answer to yourself, and if you are not able to give answer for this, then you need to sit little bit more with the complement system, okay. And last but not the least, T cell mediated disease or type 4 hypersensitivity. So cytokine mediated inflammation, CD4 positive T cells are involved, and T cell mediated killing, that is CD8 positive CTLs are required. Macrophages activation, cytokine mediated inflammation, direct target cell lysis, cytokine mediated inflammation are the major mechanisms through which type 4 hypersensitivity will take place. I guess I don't have it. I did put it though. Just give me 1 minute. Oh, okay. So I forgot to put a visual aid for this, then this whole thing would have been a little bit more clear to you guys. So what we'll do is we'll try to solve some questions, because I'm assuming you guys are up to date with the lectures. So, and discussing questions is important. So we'll start with the complement system questions itself, okay. And then I'll put some visual aids for hypersensitivity. Maybe I might have put them already in the sample questions. So the rules are same. I'll give you 30 seconds to 50 seconds for simpler questions, and more like 1.5 minutes, or like depending upon the question. And you have to type your answers in the chat box. Everyone should attempt it. Everyone should try. And even if you don't know the right answer, even if you know the wrong answers, that why this particular thing is wrong, still remember that those options you have to be very clear that where you have studied, what is the role of those options and all those things. It basically helps in summarizing the particular topic, okay. So the first question for today is, the protein which catalyzes the proteolytic cleavage of the complement protein is called. A convertase, a protease, a complement enzyme, or a complement cleavase? It's a pretty easy question, guys. So you just type your answer in the chat box. I don't think so that so it's a pretty easy question. Just type your answer in the chat box. I don't think that we need any time for this question. So we have heard this name a lot while we were discussing about the complement system, right? So a protein which catalyzes the proteolytic cleavage of complement protein is called convertase, protease, complement enzyme, complement cleavase. What? Okay, so I understand where the confusion has come. So your answer, protease. Protease means any protein or any enzyme basically which is digesting protein. It's true. But here we are talking about cleavage of complement proteins. So we are giving the specific name to the proteins for which we are asking for the enzyme. So it will be convertases. So you remember we need to make C3 convertases and C5 convertases in the complement pathway. So that was the right answer. Okay. So the next question for today is, several of the complement components are a glycolipid, cell surface molecules of lymphocytes, precursor of enzymes, hormones, or antibodies. This you have to think a little bit, but still it's a very direct question. So I'm gonna give you guys 1 minute, but it will be 30 seconds or something, and type your answer in the chat box. It's a glycolipid. Okay, so I completely understand from where the confusion might have come, but what I'll suggest you is to take any textbook, or you can go back to your lectures, go through the complement system lectures one more time, and then so the correct answer is glycolipid. I completely understand your confusion. So what I'll suggest you is, take any textbook or take any even you can go back to the lectures itself, then try to answer this question. Your question is completely fine, sorry, your answer is completely okay, that the precursors of enzymes, so several of the complement components are, but you need to understand that we are talking about the components, not complexes. So it's a very basic difference. Okay, so me explaining to you, it won't help you that much unless you understand the complement pathway well. So I'll suggest you to go back to the lectures, and then in the next class we can have a discussion on this. Okay, so the next question, the cofactor which is not involved in factor 1 mediated cleavage of bound C4b in classical pathway is C4b binding protein, complement receptor type 1, membrane cofactor protein, or factor H. This you can answer only if you have seen the lectures. You guys have 1 minute. Okay.
Option B, ma'am.
Okay. Anyone else want to give it a try?
Option D, ma'am.
Yeah, I heard you. I'm asking about other people who are also in the meeting. Anyone else? Okay. So the correct answer is D, factor H. Again, okay. If you have been introduced to the complement system for the very first time, all these things are pretty understandable because a lot of new terminologies, new names, new functions, new conjugation like simultaneously have been introduced to you guys. So sit with the complement system. The best way again is to write it down on your own, that which component comes first and then second and then third. Then you'll understand that these questions are the easiest to solve because they are just memory based, right? And the way to keep them in the memory, as I've told you, is to remember them as a movie, that what is the initiation, what is the C3 convertase, and what are the components in C5 convertases. Okay. The next question, the classical and alternative pathways meet at the complement component. C4, C4b, factor D, C5, C3. I'll read the question very carefully. So the classical and alternative pathways meet at the complement component. We are not talking about classical and lectin. We are talking about classical and alternative. Okay. So where classical and alternative pathways meet? Do they meet at C4, C4b, factor D, C5 or C3? Okay, so 30 seconds is enough and your time starts now. Anyone wanna give it a try? Okay, so the right answer is C3. Okay, those who are not able to get it, again go back to the complement lectures. You'll see they are very direct, like literally direct questions. Next question, what does the membrane attack complex in the complement pathway consists of? Colicins, C3b, B3b, BB, OH, or C5b, 6, 7, 8 and 9. I don't think so that we need any time in this, but still I'll give you 20 seconds. Yeah, so obviously the right answer is C5b, 6, 7, 8 and multiple units of 9, C9 component. The complement proteins of the lectin path. Oh, I'm so sorry. So I was saying, so in this what I said is obviously the correct answer is C5b, 6, 7, 8, 9 and especially multiple units of C9. Okay. So the next question for today, complement proteins of the lectin pathway bind to blank on the pathogen. Now here we are asking about the initiation of the complement pathway itself. Which pathway? Lectin pathway. So do they bind to proteins, phospholipids, carbohydrates or nucleic acids? You guys have 30 seconds for this question. So the right answer is carbohydrates. So what is, it's in the name, right? So lectin, it is identifying lectin, mannose and all those things, and those are carbohydrates. Okay. So of the following, which one is the most important function of the complex formed by the complement components C5b, 7, 8 and 9? First, to enhance antibody production. Second, to inhibit immune complex formation. Third, to opsonize viruses. Fourth, to perforate bacterial cell membranes. 5, to release histamine from the mast cells. Again, see, you can understand, right? If you know the answer of the complement system, they are direct questions. So it's better to remember them. Just 30 seconds for this particular question. Oh, we already have answer, right? You need to cross check once. So the right answer is D, to make the MAC, membrane attack complex. So it will basically perforate the bacterial cell. Okay. And here's the explanation for that. Innocent bystander lysis is a phenomenon in which T cell kills pathogens, complement protein C5b67 kills normal cell nearby the target cell, antibodies engulf the pathogen, cytotoxic T cells release granzymes to kill infected cells. If you have gone through the week's lecture, you will be able to answer this, but I'll give you 30 to 40 seconds for this particular question. Okay. Yeah. So the right answer is B. Complement protein C5b67 kills normal cells nearby and target cell. So as you can see, innocent bystander. So innocent means these cells were not at all harboring any pathogen, nothing was there, yet because of the complement activation and because of the chaos of inflammation and everything, even the innocent cells are killed. Okay. The next question: what are the roles of alpha and beta subunits of GM-CSF subfamily of class 1 cytokine receptors? Now we are switching our gears toward non-complement questions. So both subunits induce signal transduction. Alpha subunit binds the ligand and beta subunit transduces the signal. Beta subunit binds the ligand and alpha subunit transduces the signal. Both subunits bind the ligand. I'll give you 1 minute for this, because we have not covered this, but in the course it has already been covered. So the right answer is option B. So if you have seen the receptors, classes, the function and how they dimerizes, you will be able to answer this very well. But just to jog your memory, we'll have this visual aid and we'll see what exactly we were talking about. Okay.
What if I
talking about how immune cells communicate with one another. And where we left off last, we were discussing cytokine receptors and chemokine receptors. Now let's talk about cytokines and chemokines themselves, as well as some common naming strategies for these small soluble proteins. Recall that cytokines are used by immune cells to communicate with one another, while chemokines guide the migration and localization of immune cells. Non-immune cells can also release cytokines and chemokines to alert and recruit immune cells. Before we jump into the functions of these important signaling molecules, let's start with some basic cytokine nomenclature. Cytokines are grouped into structurally similar families and are named with a prefix or suffix indicating which family they are from, along with another identifier like a number or a Greek character. Some major families of cytokines include the interferons, abbreviated as IFN, tumor necrosis factor, or TNF, and colony stimulating factors, which have CSF as a suffix. One of the largest families of cytokines is the interleukins, whose name refers to communication between leukocytes, or white blood cells. We now know that many non-immune cells also release cytokines, so while the term interleukin isn't entirely accurate, it is still widely used. Because cytokine and chemokine names are often grouped by structural similarity and organized in order of discovery, we are going to introduce them based on similar function. Learning about cytokines and chemokines can feel a bit like being force-fed alphabet soup, but knowing how to interpret their names and having a basic idea of what many of them do will be quite helpful. Now, when we talk about an immune response, there are actually several common types or varieties that are defined by distinct cytokine and cellular profiles, and these types are tailored to fight different kinds of pathogens. Each of these varieties of immune responses are named for the type of helper T cell that orchestrates the response. We'll talk much more about these helper cells later in the series, but right now let's just focus on the groups of cytokines that tend to function together, as well as the major outcomes of these signaling profiles. The first group of cytokines we will discuss are Th1 cytokines. These are the ones that drive a type 1 or Th1 immune response, named as such because it is driven by type 1 T helper cells. The Th1 response is known for promoting cellular immunity against intracellular pathogens like viruses and some bacteria, meaning that this response is geared towards activating CD8 T cells and natural killer cells, which can both detect and kill virally infected cells, destroying the virus in the process, as well as macrophages, which are great at killing pathogens they've ingested. For simplicity's sake, we won't discuss all the cellular sources of these cytokines, just some of their broad targets and effects. Here's the lineup of some of the cytokines involved in a Th1 response. IL-2 is best known for being absolutely critical for T cell survival, proliferation, and differentiation. IL-12 activates natural killer cells, which kill virally infected cells, and helps polarize helper T cells to be even more type 1-like. TNF-alpha can cause cell death and has broad-reaching pro-inflammatory effects on many cell types. Lymphotoxin alpha, or LT alpha, is closely related to TNF alpha, and it can signal in its soluble form as a homotrimer, or in its membrane-bound form, where it complexes with lymphotoxin beta, or LT beta. Both LT alpha and LT beta can kill chronically infected cells, activate macrophages, and are important for lymphoid tissue development. IFN gamma is a type 2 interferon. It has direct antiviral activity on cells and helps make virally infected cells more likely to be detected by killer T cells. IFN gamma also activates macrophages and blocks polarization of type 2 helper T cells, which we'll talk about next. After Th1 cytokines, we will discuss Th2 cytokines. This group belongs to the Th2 response, which is controlled by type 2 helper T cells. Th2 responses activate humoral responses, meaning that the immune response is dominated by the action of antibodies, which are produced by activated B cells instead of killer T cells. It is also characterized by a strong presence of eosinophils, basophils, and mast cells. Th2 responses are characteristic of a response to parasitic worms called helminths and can also promote wound healing and tissue repair. Th2 responses are also responsible for most of the negative effects that occur with allergies and asthma. Here are some of the most important Th2 cytokines. IL-4 is critical for polarizing helper T cells to a type 2 phenotype. It promotes mast cell growth and development in the bone marrow, stimulates eosinophils, and activates B cells. IL-5 drives eosinophil development in the bone marrow. IL-13 signals B cells to make a class of antibody called IgE, which is one of the hallmarks of a Th2 response. IL-25 helps amplify Th2 responses by inducing IL-4, IL-5, and IL-13, and activates type 2 innate lymphoid cells. IL-10 enhances B cell activation and antibody production while suppressing expression of Th1 cytokines. For the third group, we will jump up to Th17 cytokines. These belong to the Th17 response, named as such because it is driven by helper T cells that secrete high levels of IL-17. Th17 responses are especially good at controlling fungi and extracellular bacteria because of their ability to recruit a strong neutrophil response. An overly active Th17 response can lead to negative health consequences like irritable bowel syndrome. Here, the relevant structures are as follows. IL-17 stimulates production of the chemokine IL-8, also known as CXCL8, which is a strong neutrophil chemoattractant. IL-22 acts on epithelial cells in the gut, skin, and lung, which are barrier cells that are right at the front lines of many bacterial and fungal infections, to promote cell proliferation and tissue healing, as well as production of antimicrobial peptides. And IL-23 helps polarize helper T cells to a Th17 phenotype. The fourth and final category of T-cell-associated cytokine responses we will talk about is from regulatory T cells, or Tregs. There is actually one more major subset of CD4 T cells, and these are the follicular T helper cells. These are the cells that help activate B cells in lymphoid tissue, but we'll talk about those in another tutorial. Back to Tregs, these work to dampen an immune response in order to prevent excessive tissue damage and the risk of developing autoimmunity. IL 10 is an important regulatory cytokine we've already discussed. Another one is TGF beta. TGF beta has a huge range of functions, including promoting tissue repair and wound healing, but it is also anti-inflammatory. It achieves this activity by promoting Treg development, inhibiting B cell proliferation, and inhibiting activated macrophages. So with cytokines covered, now let's switch gears and talk about chemokines. Chemokines help regulate all aspects of cell movement and migration, including promoting changes in cell adhesion and motility, causing cytoskeleton rearrangement, and guiding cell movement towards a certain stimulus in a process called chemotaxis. Chemokines don't stop there, though. Remember how well organized lymph nodes are? A huge part of that meticulous organization comes from chemokine signaling activity shepherding immune cells to their proper locations within lymphoid tissue. Before we get started, chemokine nomenclature can also get a little confusing, so let's break it down. There are two major families of chemokines. Those are the CC family and the CXC family. CC chemokines are named as such because there are two adjacent cysteine amino acid residues near the amino terminus of the protein. Each of these residues is represented with the letter C. CXC chemokines also have two cysteine residues near the amino terminus, but they are separated by a single variable amino acid, marked by the letter X. Here's a tip. If you find CC or CXC followed by an L, for example, CCL2, the L stands for ligand. CCL2 is therefore a chemokine. If CC or CXC is followed by an R, the R stands for receptor, and you know that this protein is a receptor for either a CC or CXC chemokine. Let's look at some CC chemokines. These tend to induce migration of lymphocytes and monocytes. CCL2, or MCP1, promotes Th2 immunity and release of histamine by basophils. CCL3, or MIP1 alpha, recruits monocytes, macrophages, and neutrophils, and promotes Th1 immunity. CCL4, or MIP1 beta, recruits monocytes and natural killer cells. CCL5, or RANTES, recruits eosinophils.
This is how the brain of a procrastinator.
Basophils to
So you get an idea about these, right? So in the lectures, maybe last week lectures, not this week's lecture, the this receptor structures were discussed, and what are the role of each monomer, each subunit. It was discussed very well in that. So for today I might stop here. I will stop here, and in the next class we'll discuss more about the, say, autoimmunity aspect of it along with hypersensitivity, and then more questions. So we're gonna focus a little bit more on the questions because the lecture will be ending in the next class. So we'll focus a little bit more on the questions, which will help you in your exam as well as in your assignments. Okay, so thank you so much for your patience. I'll see you in the next class.